All right, so I'm going to discuss the scientific thinking, which is with 1.6, the science of nature from the textbook. And our objectives for this section are going to be to describe critical thinking, differentiate between inductive and deductive reasoning, explain the independent and dependent variables and give examples of them, and as well as explain the importance of a control group in an experiment. All right, so what is critical thinking? So it's determining the quality of the information before you accept it as true. So you're just not saying, oh, because this person said that, it must be true. You're figuring out what your sources are. So how we do this is we kind of run through, and you probably do this anyways when you hear things from other people. What is the message? Is it based on a fact or is it opinion? Is it some little bit of both? Does the presenter possibly have any bias? Do I have any bias? So we tend to accept things that kind of support our beliefs. But when you're thinking critically, you're trying to look beyond that and trying to figure out what is fact and what is opinion. And sometimes they are very interwoven, so it takes some practice. I haven't posted it yet, but I will post up the how to spot bad science, and maybe I'll flip over to that, or actually I'll put that up, like I said, after I get done doing this. So it has a great breakdown of, what's the source, where did it come from, and so on. All right, so let's look at scenario number 1. So I'm going to imagine this, again, was intended to be done in class, but how many of you have placed your cell phone on charge, when you went to bed and then woke up and then realized it didn't charge? I would imagine everybody in the class has had that happen at least once, probably more times than that. So you're probably already thinking about some possible reasons why it didn't charge. And a lot of the reasons you're coming up with are going to be based on your own prior experience with this scenario. Like, what would be some reasons? So maybe it wasn't plugged into the phone correctly. Maybe the charger got knocked out of the outlet. The power went out. The charging cord is broken, has a short in it. The outlet isn't working, has a short in it. Maybe someone in your house unplugged it accidentally to plug in their charger. There's a whole bunch of other possibilities, but these are just the ones that sprang to my mind when I was making this PowerPoint. So these examples here are examples of inductive reasoning. They are conclusions based on observations that your phone didn't charge. So inductive are conclusions based on observations. So then what would you do next? So you'd make a prediction. If my phone didn't charge due to a short in the charging cord, then using a different charging cord would allow me to charge my phone. So the first one, your hypothesis is, oh, it must be something wrong with the charging cord. A prediction gets a little bit specific. So hypothesis is a little bit more generalized. Predictions tend to be more specific. You probably did this a lot in, like middle and high school, if you remember, with the if-then statements. We're going to call those more precise, so there's going to be predictions, and they're going to really zoom into what the cause and effect are. If my phone didn't charge due to a short in the charging cord, then if I change to a different cord, then it'll allow me to charge my phone. Or if the phone didn't charge because the charger wasn't plugged into the phone correctly, then pushing it in completely will allow me to charge my phone. So using your hypothesis to make a prediction is deductive reasoning. So you're making a conclusion about your specific scenario here, the specific circumstances here. All right, so then it would be time to experiment. And experiment is just that. It's testing your hypothesis. In this case, it doesn't seem like a big experiment, but that's what you're doing. You're actually testing your hypothesis, testing that prediction. So it doesn't seem like an experiment. We, probably have in mind when you hear the word experiment, being in a lab with beakers and, fire and test tubes. And it doesn't need all that. Anything where you're testing your hypothesis, your prediction, is an experiment. So if the hypothesis was, the prediction was, if my phone didn't charge due to a short in the charging cord, then using a different charging cord will allow me to charge my phone. So then the experiment would be actually switching out the charging cord. Maybe you go grab your roommates and say, hey, let me borrow this. I know we have the same phone. Or maybe you know, have a car charger that you can go and plug it in and see. So if the hypothesis is rejected, so maybe you switch out the phone cord, you go grab your roommates, plug it into the same outlet, and it's still not working. So that doesn't support your hypothesis, so you have to redo your hypothesis. Now, if it charges, great, it supported it, and you know that, okay, yeah, it was the charging cord. But if it doesn't, then you have to redo your hypothesis. So you might go back and say, let me go back to the other one. You might go back and say, you know, let me try a different outlet. Let me, you know, check to make sure the power's on or whatever. So scenario 2, same idea. So you're at home reading the next chapter in your biology textbook when suddenly the power goes out. You don't want to waste your phone battery because the power's out and you want to make sure it stays charged. So you've managed to find a flashlight. However, it's not working. So using the scientific method, and test what is wrong with it. Take a few minutes and write down a possible solution to the problem. So again, this was intended to be done in class. You can pause the video, think of some things. Remember what I said before, it's good to test yourself to see what you know, what you don't know, and keep moving on. So you should pause and then resume when you've written down a few things. Importance of a control group. So a control group, or simply control, is important to validate the results. And I just realized this won't let me go back to the other slide. I was going to go over the other thing. So anyways, the control group is used to validate the results. So maybe like with the flashlight, if you're thinking that the batteries were dead, maybe you switch out the batteries. And you're really comparing it to the other one. So with the flashlight, it's not a great comparison. So for the control, you're really just having something to validate. If I want to see if, you know, Miracle-Gro really grows my plants bigger, I need to grow some plants with Miracle-Gro and some plants without Miracle-Gro and compare them. Otherwise, I don't really know how to factor in on it. So the control group, the conditions are kept the same, except this group does not receive the independent variable. It is not tested on. This allows the control group to be compared to the experimental group. The experimental group is the group that is being tested on and receives the independent variable. By comparing, we can see if the independent caused whatever to the condition. So if I'm looking at the Miracle-Gro, if I have 10 plants that I plant with use Miracle-Gro on, and 10 plants that I don't use Miracle-Gro on, everything else should be the same except one group receives Miracle-Gro and one group does not. And the purpose of that would be to compare them to see, did the group with Miracle-Gro do better than the group without it? It validates those results, because if I just grew a bunch of plants with Miracle-Gro and I had nothing to compare to, how do I know it's not the plants were really good? Maybe they're really fast growing plants. Maybe it was really good weather conditions. Maybe it was, the type of water I use, the type of soil I use, whatever. So by keeping everything the same except one group gets that independent variable and one doesn't, I can compare and say, yes, that's exactly what had an effect. Kind of going back to the flashlight. So if you're testing the flashlight out, you're really comparing everything to what was before. So the starting point is the flashlight didn't work. Now if I take the batteries out and put new ones in, does it work? No. Then it must not be the batteries. Let me change the light bulb. Maybe the light bulb's burned out. Does that work? No. Well, maybe it's something else. Maybe there's a corroded connection. Maybe there's a wire that's loose. There's a few other things we've done there. So you're comparing it back and forth to see if it actually had an effect on it. And in this little picture, this is one I've done with my high schoolers. I do this at, other college level classes. So you take, and you make these are Erlenmeyer flasks. You take 2 flasks and you fill them up with, let's say, 500, 400 milliliters of water. I think it's 400 milliliters of water. And you add some drops, or like 2 drops of green food coloring. But to one of them you add bleach. And when you stir it, you get those beautiful color changes. It changes from green to blue. And the reason why we know it's the bleach and not something else is because we can compare it to the other flask. So everything else was the same type of container. I used tap water for both. I put 2 drops of green food coloring in both. I stirred them for, let's say, 60 seconds each. The only thing that's different is that I added bleach to the one on the left, the one that is now blue. So this validates our results so that we can say, yes, the bleach is what caused the color change. If we just had the one on the left and all of a sudden we had blue water in there, one could argue using critical thinking skills, well, how do we know it was the bleach? How do we know there wasn't something in the flask to begin with, or something in the air, or that it was just going to change anyways after a few minutes? So this actually shows us we can compare and say everything else was the same except this one received the bleach, this one did not. So the one that did not receive the bleach, the one on the right, is our control or control group. All right, so next section, this is from section 7 or 1.7, analyzing experimental results, our objectives, risks and generalizing data from a subset, identify ways that researchers minimize sampling errors and bias, talk about statistical significance, and identify areas of inquiry that are not addressed by science. So a subset is just that sub-meaning below. So this is a smaller sample size of the population being studied. So if I wanted to survey students at Norfolk State on their opinions about science, it would be very impractical to expect me I could ask every single student. I mean, I could, and that would ultimately be the best result, is to ask everybody and then put those results together and say based on all the students. But I could realistically ask 100 or 1,000 students. This number would be a subset. It's a smaller representation of that population. The more I ask, the more accurately it reflects. If I just went and picked 5 random students, or even if I picked 5 students, let's say in the science building, does that really represent what all students that attend Norfolk State University think? Would there be some biases there? If I just went to the science building and picked students, chances are students who are in the science building, especially if I went to, let's say, an upper level science class, maybe like a microbiology or something that, not every student would take, there might be some biased opinions there because they are most likely majoring in science. So maybe theirs would be favorable right off the bat because that's what they're majoring in. But again, if I just asked 5 students, even if I just walked out, to the student center and asked 5 students, that's still way too small of a sample to say that it represents all of the students at Norfolk State. So that's part of the problem with generalizing based on a small subset, is you can have very biased opinions. It doesn't necessarily represent the whole accurately. And I talked about some biases there as well. So the first part really is to try to avoid them. Is that practical in every situation? No, it's not always going to happen. But just be mindful of them and ways to prevent them. And I already talked about this, using the large enough subsets to represent the population. And then you can use error bars with your data when you're doing graphs to show the possible deviation from the average or from the mean of your information there. So you're still going to have some sampling errors and bias, and this is where the peer review process comes in. Scientists love to prove other scientists wrong. If you're that type of person, then science is for you. they will purposely try to replicate an experiment and then say, hey, wait a minute, we didn't get that. Or even just sometimes reading through their published results, they can find bias in just the language. So that helps to eliminate some of those things. Does it completely get rid of it? Absolutely not. Especially if you have people who have similar biases that are looking at the same thing, they might not recognize it as a bias. That's why it's good to have a wide range of backgrounds in science so that more viewpoints can be seen and more areas of bias can be acknowledged. This is just a quick little graph that's showing some error bars. You see they look like giant eyes, and it's just showing you, and I just did a random search for random sampling errors. Actually, I looked for error bars in the image section. So they allow for, to see how statistically significant it is. So did it occur by chance, or is there a correlation between these two events? This is talking about breaks for wool A and wool B. I don't know the whole thing. I guess they're trying to measure the tension of these different, the strength of these different wools to see, how quickly they break. And they have the mean breaks on the side. So like for the first one, it's a little over 40 for the first wool A, and then for wool B, it's just under 30, so maybe like 28, 29. But you can see the error bar gives a pretty good range there from like 20 to almost 40 for wool B, and where it's at for wool A on that far left one, it's labeled L on the x-axis. You have from like maybe 27 to like 60, let's say 2. So that's a pretty good, huge margin of error, I should say. But I just really wanted to use that to show you what the error bars look like if you hadn't seen those before. All right, this is section 1.8, the nature of science. So what is science? What isn't science? Talk about criteria that must be met for a hypothesis to become a scientific theory, discuss what happens when data contradicts a theory, and distinguish science from pseudoscience. I have an excellent video here that I was going to show in class. I would encourage you to watch this. It'll break down fact versus theory versus hypothesis versus law. The short version is a hypothesis is a testable prediction to the outcome of an experiment. It is unproven. It is unproven. However, a theory is this hypothesis that has been proven over and over and over again. There has to be substantial evidence to back it up. So in everyday language, non-scientific language, we tend to say, oh, it's just a theory. Or my theory is that he didn't really want to come to work today, so, he pretended to be sick. That's just your guess. But in science, theories are supported by evidence. They are facts. They are accepted as true because they have evidence to support them. So the thing people don't like about science is that theories can change. If we have new data to contradict it, we're going to change or amend it. And people get this in their head that science has to be the same. Well, it's a good thing science changes. Science changes based on new evidence. We're not just saying, oh, we don't like this one today. We get new evidence, we say, oh, we know more now than we did yesterday. Let's amend this, let's change this. And like I said, in the video, he goes into a few more examples. There, I do have another video on here. It's a song. It's kind of old, and I know I've shown it for my high schoolers before, and they've kind of cringed at it. But I always tell them, if you don't like it, make one better, and I'll use that one. But I personally think the song on this is a little catchy. So let's talk about miasma theory. Miasma theory, if you haven't heard of it, was this idea that bad air made people sick. So you might have seen like the plague doctor masks like this. They were thinking that it was the bad air. Now, kind of in some ways, but no. When we found out about microorganisms, when the microscope was invented and we found out that there were microorganisms, there were things smaller than we could see with our eye, and then we started to actually test this. We had a lot of scientists that ran different tests and proved that certain microorganisms carried or caused disease. Well, now we couldn't say, oh, it wasn't just bad air or bad karma or whatever. It was actually these germs. So that's where germ theory of disease comes in now. That's the replacement of it. So new evidence, new data changes what we think. You know, based on old experiences, they thought it was the bad air, but now we know that you get sick from microorganisms. And I said kind of bad air because, you know, there are airborne, like the flu each year, you know, it's airborne. COVID was, you know, it's air droplets. So it's not really bad air, but these germs actually have to get into you, and there has to be enough of them. That's a whole nother thing to make you sick. So that's the amendment or the changing from one theory to another. I pulled this directly out of the textbook. So talking about science versus pseudoscience. So it compares some, you know, basic questions. Is the claim about an observation of a natural phenomenon, does it have only observable natural world things? Is there evidence? Can it be testable? Is there data? Is there evidence? Is it consistent with other scientific evidence? Is it self-correcting? Pseudoscience just tends to pick things here and there based on very small antecedent evidence. You know, well, I have a friend that it worked for, so it must always work. Just because it worked for one person doesn't mean it's going to work for everyone. So, and a lot of times in like pseudoscience, there's like it's always right. They don't have a room for being wrong, or they kind of get dismissive, you know, or some people get angry, you know, if you're dismissive because they don't have evidence. But good science, good scientists know that we don't know everything, and we can use those critical thinking skills to build upon our knowledge base. And being wrong and finding, you know, new evidence to support different ways can, you know, is part of that self-correcting. So there are standards for accuracy and objectivity. Can the experiment be replicated? You know, that's the other big thing. If I do an experiment and I publish results, you should be able to go and replicate that experiment. And if you can't, then we need to see what was going on. Was there an issue on my end? Did I interpret something wrong? Was there some contamination? Was there some bias on my end? You know, that's where science is correcting. Whereas pseudoscience tends to say, this is the way it is because we said so. All right, and I believe that is the